Okay, everybody, we're going to go ahead and get started. So I want to welcome you to our Memory and Mobility Update. We do this every year, and we always have a wonderful crowd, and we're so delighted and really honored that you are joining us this afternoon. You are going to hear about our Discoveries and Advancements in Neurodegenerative Research program, which impacts diseases like Alzheimer's disease, dementia, ALS, and Parkinson's disease, plus many other more rare types of neurodegenerative conditions. So before we get started with the program, I want to acknowledge that tonight's event is part of our 15-year anniversary series. We've had programs like this all year long, and I want to thank the sponsors of our anniversary series, who is Evolution and the Keen Group at Morgan Stanley. They have sponsored all of our anniversary series events, and we're grateful. I also want to, absolutely, we can clap for that. Absolutely, thank you. Thank you. I also want to thank our presenting sponsor for tonight's event, and that's Twickenham Advisors. And Wes Clayton and Twickenham Advisors has been a sponsor of the Memory and Mobility event for many years, I think since we started. And we are so grateful for their ongoing support in recognizing the importance of sharing the research that's happening at HudsonAlpha. And now I want to welcome Rob Warren, who is a partner and a private wealth manager at Twickenham Advisors, to say a few words. Rob?
Thank you so much, Elizabeth. And again, my name is Rob Warren from Twickenham Advisors, and on behalf of Wes and the rest of our team, I want to welcome you all to a wonderful event. We are very fortunate to have HudsonAlpha and the incredible people and talent at HudsonAlpha in our backyard to really see and understand the impact that they're having, not just for us in our local community, but across the entire world for many, many generations to come. So we are incredibly thankful to be a part of this wonderful event, to support such a wonderful cause, and certainly appreciate the opportunity to continue to partner with HudsonAlpha going forward. So thank you very much. Look forward to the event.
Okay, without further ado, I want to introduce Dr. Sarah Sharman, who will be the moderator for this evening. Sarah is the Research Communications Manager at HudsonAlpha and also the co-host of the Tiny Expeditions podcast. And if you have never listened to the Tiny Expeditions podcast, there are 4 seasons of this, and I encourage you to go to the HudsonAlpha website and check it out. I promise that you will really enjoy listening to all 4 seasons. So with that said, I'm going to turn the microphone now and the program over to Sarah.
Thanks, Elizabeth. Tonight, it's my pleasure to moderate this exciting discussion about the amazing work our M&M program has helped make possible over the years. I'm going to ask our panel a number of questions a lot of you might have on your minds about neurodegenerative diseases and the research going on here at HudsonAlpha. In fact, some of you even submitted some questions when you registered that we're going to get to later. First, let me introduce our panelists. I'm joined tonight by Dr. Rick Myers, HudsonAlpha's Chief Scientific Officer, President Emeritus, and M.A. Loya Chair in Genomics, Faculty Investigator Dr. Nick Cochran, and Senior Scientists Brian Roberts and Dr. Lindsay Rosardi. They are each experts in genomics, specifically how genomics plays a role in neurodegenerative diseases. As many of you know, genomics is an important and growing field at the core of HudsonAlpha's mission. Each one of us carries within ourselves a unique set of genetic instructions that determine our physical traits, susceptibility to diseases, and even our responses to various medications. Unlocking this treasure trove of information allows us to gain profound insights into the inner workings of our body and unravel the intricate genetic code that shapes our lives. Genomics enables us to identify and understand the genetic factors underlying numerous diseases. It helps researchers pinpoint genetic markers associated with increased risk or protection against certain diseases. This knowledge can transform the field of diagnostics, enabling the early detection of diseases and personalized treatment plans tailored to individual genetic profiles. Genomics represents an extraordinary frontier in human health research. It can transform our understanding of diseases, personalize medicine, and empower individuals to take charge of their health. Let's kick things off and learn how Hudson Alpha is using the power of genomics to improve the lives of individuals living with neurodegenerative diseases. Rick, we'd like to start with you. Can you please briefly explain what neurodegenerative diseases are?
Notice she said briefly, so I'll try. But those of you who know me laughed. But we have about, I don't know, several trillion cells in this part of our body from here up. Our brains are really complex. They're composed of a whole bunch of different kinds of cells. You've heard of neurons, but there are many other cell types. And a neurodegenerative disease is one where some of those cells die. And unfortunately, when they die, even if it's just a few cells, they start to make other cells die. And so all of the neurodegenerative diseases, and there are at least 600 of them, fortunately most of them very rare, but some of them quite common, the hallmark of those are that you have a loss of cells. And as the cell loss increases and you lose more and more, then you start to have symptoms from those. Probably are having some cell loss even before the onset of symptoms, not probably, definitely, before the onset of symptoms.
Can you describe some of those symptoms of neurodegenerative disease?
Well, they are varied, and some of my colleagues here can probably do a better job. Generally, we call this the M&M Fund. That was named by Elizabeth, Memory and Mobility Fund. And what that really refers to is that you often have, in these diseases, not all of them, and it may not be the only symptoms you have, but you have loss of memory, sometimes gradual, then it gets worse and worse, and sometimes loss of mobility, or it affects mobility in some way or another, and sometimes in a very big way. But the truth is the symptoms vary. You even end up with dementia in some. In other cases, you don't. And so they're quite varied depending on the disease. And sort of related to that question is you might have different symptoms, but there's some similarity amongst all of them. And one of those is cell loss. But even some of the mechanisms, some of the things that we look for often overlap in these disorders. That's true in neurodegenerative diseases, other neurological diseases, and even psychiatric disorders.
Can you tell us what neurodegenerative diseases Hudson Alpha is working on?
So Alzheimer's is the big one. That's more than 6 million people in the U.S. have it, so that's a big one. From many, many years, got interested early on in Huntington's disease. It's another neurodegenerative disease. That's a serious one that we work on, a lot rarer than Alzheimer's, but still 50, 60,000 people in the U.S. have it, a lot more at risk. Parkinson's disease, one that's more common than Huntington's, and in fact, it's not like Alzheimer's but quite common. And my father had Parkinson's disease. This affects many, many, many people. ALS, which we used to think was incredibly rare, but now because of diagnosis and understanding, it's still rare, but it's still, I know a lot more people with ALS than I did when I first started working in human genetics because it's clearly there. And then there's some others as well. Nick, you want to just comment a little bit on some of the
Another
others?
big category that we study is a basket of neurodegenerative diseases that are categorized under frontotemporal dementias, and they have some similarities with ALS, some with Alzheimer's disease, so that's another big focus area as well.
Let me mention one other thing that seems like it might be a little off to the side, but Dr. Greg Cooper is one of our faculty members, and he, working with a lot of us, has been sequencing the genomes of kids especially, but a lot of adults as well, with unexplained diseases. They're undiagnosed. We don't know what the cause is, and many, many of these, especially in the kids, are neurological. They have something to do, and autism falls under that, but many, many others. And so many of the kids, these are syndromic in that there are a lot of different symptoms in them. They're often rare diseases, but some of those are neurodegenerative too. A lot of them have seizures. If you have really large numbers of seizures, you can lose brain cells from that too. So that we put that into this same category, even though it's a little bit different, and it's a big impact. It affects a lot of people in the world.
So you described this progressive loss of brain cells and these really devastating symptoms that people get. What makes these diseases so insidious and so hard to study?
So I think one thing about these that's different from a lot of other diseases, we talk about, we use a lot of battle analogies a lot of times with diseases. You're gonna take action and fight against a heart disease or a cancer, and sometimes you win. And no one wins in a battle against neurodegenerative disease. There are some examples of some decently good therapies in some cases. Many times there's not. And that's what I think is so devastating about these diseases. And that kind of goes to the underlying disease process. These diseases start, the biology in the brain starts decades before you manifest symptoms. So that means that there's a lot of damage that's already been done, and sometimes that may be too late to stop it. Now we're getting better and better at catching things early, and that'll be probably something that we'll talk about as we go on throughout the evening.
I'd like to say no one wins yet.
That's a great point, Rick. So as Elizabeth alluded to earlier, we are celebrating our 15th anniversary here at HudsonAlpha. So we've had many successes in our neurodegenerative disease research program over the years, thanks in part to those of you who have sponsored the M&M program. What insights has the field already gained from genetics that has significantly contributed to our understanding of these diseases?
Let me start that. So we started a study on ALS 7-8 years ago, maybe a little longer ago than that, and it was the largest one of its type at the time, and it's grown to be much, much, much bigger. We discovered 11 genes that, when they're mutated, when you have a mistake in the gene, that can lead to the disease, and some of those were out-and-out causes. They weren't just risk factors. And two of those genes code for proteins, and the proteins are targets of drugs. So drug companies and various other people are working on using those two, as well as some other ones that have been discovered, as targets for drugs. And this is, I think we'll talk a little bit more about this later, but one of the big things about doing research is that you need to know what you are going to attack. If you're treating cancer, if you know that this cell is wrong and you know why it's wrong in cancer, you design a drug that goes after what's wrong. We barely knew anything about brain genes and brain disorders, and the last decade has shown a lot of growth in that. So part of that is finding what goes wrong, understanding that, and then what can you do about it based on understanding that. And that's why I said we're not winning yet, but we will. I mean, drugs are starting to be, and the knowledge base for this is growing so much and so fast, thanks to folks like this, that there's a lot of room for optimism, I think, in spite of the difficulties.
So a lot of places are doing research on neurodegenerative diseases. What unique role is HudsonAlpha playing in the field?
So, it's maybe difficult to see this when you think about, well, there's people all over the country at these major medical centers. How are we unique in this? But the reality is that scientists do get into fairly specialized areas. Researchers in neurodegeneration is a fairly specialized area. There's several hundred maybe labs that are really seriously attacking this issue full time. There is a similar composition of labs in genomics and applying high throughput approaches in functional genomics in the lab to really understand how the genome works. If you take the intersection of those two areas, it becomes a very small club. And HudsonAlpha is in that club of people who are applying these really new genomic technologies to understand neurodegenerative diseases. So we're in a very unique group of researchers in applying these kinds of approaches to understand these.
If
I could add a quick statement as well. I think Hudson Alpha is really unique because of people like you guys and this fund and the support that we have here at Hudson Alpha, because it allows us to move quickly and be really versatile when new technologies become available, that we can then apply them to the questions we're already asking in neurodegeneration. And so we have that flexibility and that versatility and the expertise that we're able to have together here at Hudson Alpha that can really take advantage of new things in the field, new knowledge, and new technologies.
Yeah, and I'll just brag on Lindsay a little bit and say that she led an effort that we published just recently that was the first ever study in the world to apply a new genomics technology to Alzheimer's disease. So that's just an example of how we really are leading the way on a lot of things in this field.
I just have to add this to actually congratulate and thank you all and many, many others. I was at Stanford for 16 years, great university. We had good labs, et cetera. I never once interacted with the public there. They just, maybe they support Stanford, et cetera, but it's just, you don't have that community connection. I'm sure some people do, but that was one of the appeals about coming here. While I grew up in Alabama, I didn't know Huntsville very well at all, and from even before we got here, it was very clear that, and many of you in here have really helped us get started. And the reason what was attractive to us, and I think about getting us started, was that we had expertise in this area of high throughput genomics. We sequence genomes here. We've been involved in that since the beginning of the ability to do that. And that's still, we're not totally unique in that. A lot of people do it, but I still think we do it, and I'm bragging on others, do it better than anybody else. I'm not bragging on me, I promise.
So continuing on with our discussion about Hudson Alpha using new technologies, Lindsay, do you want to talk about some of the new technologies that we're using here to answer big questions?
Yeah, of course. So as Nick mentioned, we've recently applied single-cell technologies to interrogate Alzheimer's disease. And so, as Rick mentioned too, you have so many different cell types in the brain, each of which has a different response to neurodegeneration and to the things that are causing neurodegeneration. And so to really understand what's going on, we need to figure out what each cell's response is and which cells we can go in with drugs or other targeted therapies to modulate that response so that we can either preserve the neurons that are remaining, prevent neurons from dying, or do other things with other cell types to support those remaining neurons so they can function better and make new connections. And so using single cell technologies, we're able to look at individual cell types, ones from healthy individuals and ones from in neurodegenerative cases in Alzheimer's disease, and see what the differences are in each cell type and the genes that are dysregulated or not functioning properly in each cell type. And with the multi-omic technology, we're looking at two different modalities so we can identify the gene's expression and we can identify the regions of the genome that could be regulating that expression. And so this also informs for understanding disease risk, because when we have mutations in these regulatory regions controlling genes we know are involved in modulating that neurodegeneration process, we can then better understand how an individual's risk is altered by having mutations in those regions. And this is a really active area of research that we're really starting to lead the way on in not only identifying the regions, but then also validating them functionally and saying for sure that yes, this mutation in this region is causing an effect in this cell type, and then how that's going to affect disease progression and onset. Other technologies that we're using to do that validation is using cell line models of neurodegeneration. So we're taking cells that are pluripotent, they can differentiate into many different kinds of cells, and we're differentiating them into neurons or those support cells, those other cells in the brain, and then making mutations in them to see what effect they have on function. We're checking to see, do these regions of the genome actually affect the function of these genes that we know are important for Alzheimer's disease. So we're kind of merging two new technologies together to really gain an understanding of what's happening in neurodegeneration so that we can design better therapies and move the field forward.
And if any one of you ever want to see a photograph or even through the microscope, these neurons these are neurons generated from human cells, usually skin cells or sometimes cheek cells, that you turn them into neurons and they're beautiful. You have to come see them in the lab sometime.
It's like science fiction, huh? So one of the things that impressed me when I came here to work at Hudson Alpha was just the level of collaboration, not just here within these walls, but elsewhere in the field. Can one of you kind of touch real quickly on how our work is complementing other research initiatives and other research institutes?
Absolutely. So I think there's two different ways that that happens. One is there are times when we can't do it by ourselves and we need to work with other groups that maybe bring something to the table, like a really unique group of patients, and then we bring the genomics expertise. So that's one example. And for example, in the past couple of months, we've published a really important study on a large family in South America with a rare dominant form of Alzheimer's, and looked at genetic associations with their age of onset across 340 individuals in that large family. So that's an example of where they had this very unique cohort. We had the genomics expertise, and we came together to do something collaboratively. There's also occasions when our work can be of value to the larger field on initiatives that maybe are led by a different group, but that are very important. And back to what Rick was talking about identifying key genetic causes. For example, in the past year, we published collaboratively with a large international consortium a paper in Nature Genetics where we described 2 new risk genes for Alzheimer's disease, bringing the list of burden analysis genes. This is a rare variation analysis from 3 to 5. So that knowledge is going to continue to give benefits for the field as we study those genes in the future. Now, we were one contributor of many around the world to that study, but it's an example of how that research, we may do something initially, but then it continues to provide benefit for the field in years going forward.
So if you all were at our M&M update last year, you heard from one of our collaborators, Peter Walter, about a collaboration that Rick's lab has. Brian, would you like to provide us with an update on that project?
Yeah, sure. Ooh, that's loud. I think probably a lot of you were here last year, but in case you weren't, I'll give a little background on what ISR is. ISR is a process that is operating in all of the cells in our body, and its job is to monitor the machinery that the cell uses to make proteins. And for the most part, that's really a good thing. Making proteins is how cells get things done that they need to get done. But what recent research has led to us understanding is that ISR might be a little picky. It might be a little bit too worried about how well the cell's making a protein. And that's bad because one of the things that can happen when ISR senses proteins aren't being made right is it can cause the cell that's making the proteins to die. So the idea is it's better for the cell to go away than to continue to make proteins improperly. Well, in neurodegeneration, a hallmark of many different types of neurodegenerative diseases are making proteins improperly. So what ISR can do is cause neurons to die in response to those improper proteins being made, and neurons can't grow back. So when that happens, you have lost that neuron forever. If that continues to occur, then symptoms will develop in the patient that's occurring in. So Dr. Peter Walter, who was here last year, discovered this pathway. Unfortunately, he also discovered a drug that can turn that pathway down. And so instead of it being too picky, it can make it more permissive of protein being made slightly wrong. He's continuing to work on that. We actually visited him a few months ago in California. He started a new institute that's dedicated entirely to this research. And there's also a big pharma company who is developing a drug to that pathway right now, and they are trialing it in ALS patients. And the early reports from that trial are that it's a very safe drug. So at first they give drugs to people who are healthy that don't have any diseases. Those people have reported absolutely no problems with that drug. What's even more exciting is that some of the very early results are showing that it may actually be improving and slowing the disease progression in ALS patients. That is huge. ALS has no effective treatments currently. There's no treatment you can give to an ALS patient that slows the progression of the disease. And this drug appears to be doing that. So that's really exciting. What's even more exciting is there's reason to believe that the ISR pathway is operating in other neurodegenerative diseases. So if this drug gets approved eventually in ALS, and it may not, early results in clinical trials oftentimes don't work out, but this one is showing some promise, then that drug will be also investigated to be used as a treatment for other neurodegenerative diseases. So it's an area I'm really excited to be involved in. So here in Rick's lab, in just the last year, we have engineered cells with a variant in them that makes them have too much ISR activity. And as Lindsay was describing, we can then differentiate those into neurons that are recapitulating aspects of the human brain and actually treat those cells with that drug or similar drugs to it, and use that treatment to understand how that's changing that pathway and how that's changing those cells. And that could lead to us being able to find a test to determine if someone's ISR pathway is too high, and if that patient might benefit from being treated with the drug. It could also perhaps nominate new targets in that same ISR space that could be targeted with drugs and also offer as a treatment for neurodegenerative diseases.
Thanks for that exciting update, Brian. During the introduction, I briefly mentioned that genomic information can inform genetic risk, early detection, and treatment. I'd like us to cover each of these 3 topics. So we'll start with genetic risk. Rick, can you briefly describe what genetic risk is?
Sure, although I think Nick can do a better job, but I will. When something's genetic, it generally means you were born with a change in your DNA that either causes something or increases your risk for something. So genetic risk, you can have, for a lot of diseases, you have one gene with one spelling mistake in it, and you get the disease. It's all or none. Huntington's disease is sadly one of those. And there are single genes and mutations that cause rare forms of Alzheimer's, Parkinson's, ALS, other diseases as well. But then it turns out that you also have symptoms or diseases that are actually genetic. They kind of run in families, but it's not one gene. It may be 5 genes, it may be 20 genes that in combination. We've been studying these for years. They're a lot harder to study than single genes in terms of even thinking about what you do about it. But it's a major, major step forward for understanding what any of the players could be in terms of cause of disease. So that's, I think, what, Nick, anything to add to that? I don't know if I covered it the way.
Yeah, I'll add a little bit. Just, I think you covered most of it. So for anything in genetics, we think about this spectrum from a very early causative mutation, and Rick described that there are some of those for neurodegenerative disease. You often see this in childhood disease, for example, to something that's very polygenic, or it may be caused by 100 different factors that sum. So height would be an example of that, where we actually have a really good handle now on genetically what determines height, but it's 100 tiny little things. And most neurodegenerative diseases...
And Brian got a whole bunch of those.
Right, yes. So most neurodegenerative diseases fall in the middle in what we like to think of as an oligogenic inheritance model, where there may be a few strong factors, but there's still risk factors. One example of this would be ApoE for Alzheimer's disease. Now, in the past, we've always said, don't run out and get a 23andMe test for your ApoE status, but as we'll get into, it turns out that may not be such a bad idea anymore because it's helping to stratify people for treatment. But the bottom line is there are factors like that, like ApoE and some other large contributors that are probably explaining most of the risk when Alzheimer's, for example, runs in a family.
Great, thank you. We're going to turn to an audience question now. How can knowing about disease risk as early as possible make a difference in treatment?
Any one of us? Yeah, why don't you start and talk about that?
Yeah, I mean, I think it's been mentioned already, but a key aspect of neurodegenerative disease is that it starts, the processes that eventually create symptoms start early. And so people would want a way to detect those before they're still treatable, probably. And If you know that you're at high genetic risk for developing a neurodegenerative disease, that might put you in a category that would be monitored more often with tests that could detect those very early symptoms, or not really symptoms, but early markers that are going to lead to symptoms. And you can imagine that that would enable you to get insurance to pay for testing more frequently and earlier in your life. And that would be, I think, a strong motivator to get genetic testing.
Let me add something to that. Nick or Brian said that once the cells die, they don't come back. But if you could detect early, before the cell's dying, maybe they're having some illness within the cell, like ISR or something like that. If you could detect that early, it'll certainly help in development of new drugs. Because instead of treating when it's already too late, you treat earlier, the chances of a drug working are much, much better. So we call our dream, and a lot of you have heard us talk about this, and we have worked on it, is to have a blood test. You take a little bit of blood, and then you know that someone maybe way before symptoms start is developing it. And we don't really have good drugs now. They're starting to be developed, but this will help in terms of getting new drugs. So we call it's essentially early detection and the idea of biomarkers. But they also serve another purpose. The idea, if you had a blood test, even if it's not detecting real early, if there's something floating around your bloodstream because of the cells dying in the brain, and it may be even specific for a given disease, you could test for that while you're doing while you're doing a clinical trial. This is what we have to have new drugs. This would be one of the ways, then as a way of monitoring, is it working or not? Because right now, whether something's working or not are usually behavioral, memory, I don't know all the tests that are used, sometimes imaging and things like that.
So one thing to add to that, we keep saying, the neurons die and they never come back. I think that sounds pretty scary. It sounds scary to me. One of the things that's important to keep in mind is that the human brain is remarkable in its resilience. So as the neurons begin to die in a neurodegenerative process, it can take a huge amount of the cells to die before the patient has any symptoms whatsoever. So there is time to act. If you're able to detect that very early loss of neurons, and it doesn't have to be, I'm detecting 1 or 2 neurons lost, because your brain can handle huge numbers of neurons being lost. And then if we can intervene early, there really is a chance to slow the progression or stop it.
Yeah, I'll just add a specific example of that. So in Parkinson's disease, this is one area where for some patients and for some disease stages, there can be some successes. So for example, by the time someone develops a rolling pill tremor, often the first symptom of Parkinson's disease, besides maybe sleep disturbances that happen years before that aren't recognized as a symptom, but a clear symptom, a rolling pill tremor. They've lost 70 to 80% of their neurons in the substantia nigra pars compacta, which is the region of the brain critical for driving and where symptoms begin. However, with symptoms, someone can present to the neurologist. If they're a good candidate for deep brain stimulation, they can obtain treatment. At times, that can keep someone stable for a really long time, for years. And that's with that 20% of neurons that are remaining being recruited to do the job that was done by that 100 initial percent. So while it is scary that these diseases start and we may not catch them until there's been a lot of damage done, there is hope for treatment. And catching it earlier is always better.
That's why I'm optimistic. I think we're gaining more and more knowledge like that as every year goes on, and it's actionable now. I mean, it really is happening.
Great, thank y'all. Nick, you briefly mentioned your really cool South America project earlier. Can you explain that a little bit more and kind of get into why it's not only impactful for that community in South America, but really for the whole dementia community?
That's right. So what I mentioned before was a study specifically in a rare family from Colombia that has this very early onset form of Alzheimer's. They develop Alzheimer's in the late 40s. That's been an important project and we've been working with that group for 6 or 7 years now. So that's come to fruition recently, but it's grown to an expanded effort across South America 6 countries, 10 enrollment centers. And so we're looking at families with Alzheimer's and frontotemporal dementia from across South America. Now, why is that important? First, it's just important to understand these diseases across the world. And the other thing is that it helps us understand diseases that are happening here in the United States as well. So many times there are shared or unique contributors to disease. It can help us to map exactly and pinpoint what genetic causes are in all of those populations. And finally, it can help us identify new genetic causes for disease that we can track down that will be applicable. As an example, the one that I talked about earlier that we published in the rare family from Colombia, we looked at replication of the genetic signals that we identified in cohorts in the United States and in both early onset Alzheimer's disease and in late onset Alzheimer's disease. And we found shared signals between all of those different groups of patients. Those are going to be really important signals to study further in the lab, and I fully intend to do that with my group so that we can better understand the relevance not only for those rare populations, but also for the later onset sporadic populations as well.
Are you working on anything closer to home related to genetic risk or identifying new genes?
Absolutely. So, we often get the question, well, what can I do? What's going on here in Alabama? And so we've got a couple of ongoing efforts. One, we just published a second paper from a cohort of 100 families with early onset or atypical dementia that enrolled through the UAB Memory Disorders Clinic. In addition to that, we have been working on a pilot Alzheimer's disease research center. This is a really important designation for Alzheimer's centers across the country, and just submitted a grant for a larger, more extensive version of that, where we're the genetics part of that study. And so that's right here in Alabama as well. So both of those efforts are local and demonstrates that we're really trying to do everything we can to impact the community here.
So Rick earlier was talking about a blood-based test to look at whether a person was at risk for neurodegenerative diseases or whether a treatment was working. We call those biomarkers. Brian, would you like to give us an update on what HudsonAlpha is doing in the biomarker space?
Yeah, so our focus right now is actually a blood-based biomarker for ALS. We're doing this in collaboration with Crestwood, which runs an ALS clinic that services a very large area around here. So we're able to get enough patients in and get blood samples from them so that we have the statistical power to find something meaningful. And very recently, we've processed those samples, and although it's a small set of samples and we are expecting to get more, the initial results are very promising. We're looking for nucleic acids that are free-floating in the plasma. One of the nucleic acids we're finding is actually only normally found in muscle cells. And that fits in and makes a lot of sense because ALS is a neurodegenerative disorder, but when muscle cells become uninnervated, meaning they lose connection to the nervous system, they also start to die. And so to see markers of muscle cell death in an ALS patient and not in controls is pretty exciting. It's also confirming results that other people have seen as well. And that approach that we're applying to ALS is something we want to expand to other neurodegenerative diseases as well, and hopefully see similar success.
Let me mention one problem with this is the hardest part of this is not the science part that we're doing. It is collecting the appropriate samples that are clinically verified and collected in the right way. It's really just blood, but it has to be processed properly. We really want to do this in Parkinson's, and so we're setting up to do that because it'd be really useful to have something that's an early detection as well as a monitoring system. And even with the advancements that are being made in Alzheimer's and ALS, in terms of drugs, and even Huntington disease, where we know the cause is one gene, still having a blood test would be really, really helpful for, for being able to detect this, and then to use that for monitoring and earlier treatment. And so we've had numerous rounds of collecting samples. Crestwood's been wonderful about this. We're setting up to do this in Parkinson's especially, and we've done a little bit in Alzheimer's as well.
Yeah, and just, the precedent for this is it's worked in other neurodegenerative diseases. Not this exact same approach, but in Alzheimer's disease, there are protein-based biomarkers that are being used to guide clinical trials as we speak. And that didn't exist 4 years ago, 3 years ago even. And so it's a space that a lot of people are looking at, and it's exciting to contribute to that space as well.
So we will move on to treatment now. So we have an audience question, which is, why don't we have more drug treatments available for these diseases?
Yeah, so I think one of the key points for that is just there's a lot of heterogeneity in these diseases. You'll have, Parkinson's patients present that also end up with dementia, and you'll have a range of symptoms that cross all of these different kind of defined diseases, right? So that's part of it, is getting the right treatment to the right patient. And so when you have a drug that is going to, affect one aspect of the disease, if you don't have the right patients in that trial, that is the driver of their disease phenotype, then you may dilute the results that you get because you have a bunch of other people that aren't responding to that drug. Because even though they may have shared symptoms, the underlying cause of those symptoms is variable. And so this is where, again, these biomarkers can come in to kind of help us stratify these patients, not only get them in trials earlier, which we've already discussed is really important, because I do think in neurodegeneration especially, there's a threshold, and this has been shown, and I think it's a widely held belief in the field, that once you cross a certain level of degeneration and neuron loss, there's not enough that you can do, at least singly, to kind of delay or recover for those symptoms, right? And so it could be that we need definitely earlier detection to get these treatments to people earlier. There may need to be combination treatments for certain individuals because they've got multiple things going on, and no one pathway modulation is going to be effective. And so we're getting there. We're getting there with genetics. We're getting there with biomarkers. And that's why we're starting to see success in some of the trials for Alzheimer's disease and so from other things that I think we're going to talk about a little bit more here in a little bit, is because we're just now have the tools and the ability to really get at, for every patient, what's causing their disease and how to tailor things to them, get them in the right trials.
Could I ask Nick, one of the things about the Columbia or the South American studies in general is when you have a population where it is homogeneous, they all have the same cause, and get around the problem that Lindsay just mentioned about heterogeneity. Isn't that a good population for drug trials? And are they doing that? I don't know.
Yes, it is, and they are conducting trials in that population.
So, we're talking about there aren't many drug treatments, but I think there are a few. Brian, do you want to talk about any treatments that are on the market or that are in trials that are looking promising?
Yeah, so I already talked about the ISR inhibitor in ALS. Nick can chime in here too. To treat Alzheimer's disease, there have been a class of drugs that treats a protein called amyloid beta. And those have, you've probably heard about it in the news. Some of them have been controversial in that they're incredibly expensive and they don't appear to have a huge benefit to patients. There is a newest generation of that's being developed by Eli Lilly, and their clinical trial results are actually pretty striking, and that they're considerably better than the other two drugs that are currently on the market. So there's a lot of excitement for that drug to come on the market as well. And really make a difference in the progression of disease in Alzheimer's disease.
Yeah, and that's that's something that's changed just over the course of I see a lot of familiar faces here, just over the course of the past few years. So you may have remember me or Rick saying we think that this is going to get there eventually every year. for the last 5 or 6 years. And now we're there. So it's actually a really exciting time for the field. You know, I've been attending, you know, these scientific conferences for the last dozen years or so, and we'll see these presentations about phase 1 trials with amyloid therapy, and then it turns out the phase 3 ends up not working, and it's been so frustrating. But every time that there's been a failed trial, as a field, we've learned something important. We've learned about how to better control for the heterogeneity. We've learned how to treat earlier. We've learned about the type of species of amyloid beta. And let me also say, amyloid beta is a genetic target. So one thing that we haven't really explicitly said is that some of those early families, those are all mutations that are involved with processing of amyloid beta, and that applies for the entire population, not just those early cases. So that's why, you know, we may study. You hear us talk a lot about the early onset disease we study. It's because it's very relevant for the general population as well. Now, as Brian mentioned, you know, we're really to amyloid therapies that are having an effect. Now it's maybe a 25 to 30% slowing in the rate of progression. So it is not a cure, but in combination with the other existing therapies that are on the market, you will see a tangible delay in things like nursing home placement and activities of daily living decline. So they will have real benefits. You know, I won't go too far further because I think we want to talk about, you know, our optimism for the future too. But I think our optimism for the present is there, and it's very tangible for me, and I hope that you feel that as well.
So I want to add something to this. So, and this is a great example. Many drug trials are going after amyloid beta, which is actually the plaques in Alzheimer's disease. So it's the same target that the drugs are going after, and it makes sense for the reasons that he just said. But one of the mantras, and Brian could probably talk, all of the three of these could talk more about this, is that we need new targets, things that we don't know are involved yet. That's where it's not really basic research, but the exploratory research, we're trying to discover what molecules are different. And it's not just going to be beta amyloid, it's going to be, we already know some of the others. Those two ALS genes that I mentioned are part of drug trials. Those were completely new things that, because of our discovery, now we should start to make drugs after these. So think about that with regard to all of these diseases, and the more shots we have on goal, the better chance we have of making it. All of us are doing research to try to figure out some of those new ones, new targets from some of the things that Lindsay and all three of them have talked about.
So we've been talking a lot about treating with drugs. So we had an audience question asking if there is research being done on the prevention of brain disease through lifestyle changes like diet, exercise. Can any of you comment on that?
Well, certainly that research is being done in the field as a whole. At HudsonAlpha, not so much. Our focus is on genetics and cell culture models that we can manipulate artificially. Those diet and exercise and other behavioral aspects of Alzheimer's risk really need to occur on people. And so a lot of that is happening at medical schools where they have access to patients and they can do those kind of behavioral studies. It's very important research. It's just you need to have access to human beings that you can actually analyze and get data on to do those.
Large numbers of them.
Yeah, and I'll add to that a little bit. And so, we do get this question a lot because people want to know, especially, if you are affected yourself or have a loved one affected by these diseases, is there anything we can do? The lifestyle changes seem very tangible, right? Well, I could make some lifestyle changes. It really is probably also in the bucket of needing to be something that's preventative and done over a long number of years. What we know about the underlying causes from epidemiology is that 60 to 70% of most neurodegenerative diseases probably have a genetic etiology, while about the remaining 30 to 40% is environmental or lifestyle. And what's being done to test if lifestyle interventions can have an effect, that's being done very systematically across the world in a controlled way. There's the study called the FINGER study. The emphasis is on proper control of blood pressure, of diet and exercise. You're welcome to look it up if you want to see exactly what those things are. They are going to be different across different areas of diseases, though. There's maybe some signals towards a mild protective effect of exercise and dementias, but then in ALS it's actually detrimental. So you'd have to be careful depending on the disease. But generally, what the findings so far are small signals, but the signals that they do find is things that your general practitioner is already telling you: don't smoke. Don't drink or drink very little, Mediterranean diet or derivative thereof, and keep your blood pressure in check. So nothing really groundbreaking there. Just take care of yourselves.
Great. So how is HudsonAlpha contributing to this lack of treatment? I know we do a lot of basic research and we're not in the market of making drugs, but how are we contributing to that quest?
It really is the main thing. Well, the biomarkers, et cetera, we hope will make a big difference. But finding the causes, that helps the whole research community then apply those. And I think at least the one I'm hottest on, is new targets for drugs, what we were just mentioning. There are probably others as well, but that's where, and we're not the only people in the world doing this, but we do have some, what Lindsay, the study she just led, really was the first of that type, even though other people are doing single-cell genomics. It was applied there. Those are the things that we're doing, trying to do in a big way.
Sorry.
I was just going to say as well, a benefit that we have here is that we're studying not just one neurodegenerative disease, but several. And so we can look and start to see the commonalities among these and the pathways that are shared, like Brian is probably about to just talk about now. And so maybe we can find an intervention that works for one may work for others. And so that's information we can share with the field, such that, if an Alzheimer's drug comes out that is really effective and it's hitting a pathway that's also affected in Parkinson's, maybe that drug that's already going to pass trials potentially can now just be ported over to Parkinson's patients and see if that can help. And that can speed up the finding of new drugs for different neurodegenerative diseases.
Yeah, I think there's a question coming up. Spoiler alert, I saw the questions ahead of time. But before I came to HudsonAlpha, I worked for Merck for almost 10 years. Merck's a big pharmaceutical company, in case you don't know. And Big Pharma in general has made a strategic decision to focus on their expertise, focus on what they're good at. And that is the chemistry behind making drugs, testing drugs in animals, and then eventually in humans in clinical trials. And other maybe somewhat boring things like how you formulate drugs into pills or other things like that. They are not doing the basic research on what to target with drugs. They are relying on places like HudsonAlpha and other academic institutions to do that research. They read the papers that we write, they look at the targets that we nominate, and then they follow them up internally. And so without that basic research, that pipeline will dry up very quickly. And so it's an important role that the nonprofit academic research is playing in that process.
And we keep telling them they should be supporting our research. Seriously, they get great benefits from the fact that they used to do research like this 30, 40 years ago, but not like this, but they would do the basic research, and now they're relying on academia and biotech.
So we've talked a lot about clinical trials this evening, and we had an audience question. Are there any local clinical trials that we can participate in the area?
Yeah, so the answer to that, there's some good resources for this. So, for example, the Alzheimer's Association has a resource on their website. I know for Alzheimer's disease clinical trials and probably for other neurodegenerative diseases as well. If you're interested in this and you're you or a family member being seen already by a neurologist, please do ask about it. The options around here may be a little lighter, but there are definitely lots of clinical trials ongoing at UAB and Vanderbilt that could be options and sometimes don't require going in all that frequently. I would say also that, especially for Alzheimer's disease, there are some clinical trials now where you do not need to have symptoms. If you have a family history and meet certain criteria, and they may do like an imaging scan, then you may qualify for a clinical trial. So it's worth looking into. On that basis, we've been talking about how important it is to catch things early, and sometimes we can catch those things early if you know what to look for.
This has all been such a fascinating update, and a lot of this work is funded by the M&M program and people in the crowd here. So Rick and Nick, can you guys just comment briefly on what an impact the M&M program has had on your research?
Yes, it's made all the difference in the world, seriously. The way scientific research gets funded in nonprofit organizations, universities, and groups like us is you write a lot of grants. We spend a lot of time writing grants, and we get a lot of grants. NIH, but various other funding agencies, a lot of federal ones there, some private agencies. Nick has had at least maybe one or two grants from private agencies for Alzheimer's diseases. These are really important, but they don't pay for the whole thing. So this has been absolutely critical. And for another reason too, it's not just the money that's needed. And by the way, most of that money goes towards supporting the people. I mean, it takes people working really hard, and they do work really hard, and we think we're really great stewards of this. The money goes into that and into the materials that you have to buy to do the DNA sequencing, for instance, to do all the research. And so it turns out to be very important. And one important aspect of that, and this is true across the board, we get to try things that are a little riskier, not to you or to patients or people, but riskier in terms of whether they're going to work or not in our research arena, because we have funds from philanthropy that support the research. Whereas when this may sound a little cynical, but when you write an NIH grant, you have to be pretty darn sure it's going to work for it to get funded, which is a little bit of a shame, because the reason we make advances, yes, you've got to just go steady as she goes and keep going and doing this, but you also got to try new stuff. And so that's one of the things. I mean, when you ask what's good about HudsonAlpha, we do have the ability to do that more than I was able to at the university, for instance. So that probably is, maybe add
I was just going to say the single cell study that we've been talking about is a direct example of that. That work was funded by the M&M Fund, and that work has made a difference and been a leading a publication in the field. And so that is something that, that technology didn't exist when we would have written the grant. We had planned to do that project in a slightly different way, and the technology changed, and so we changed with it immediately. And so we were one of the first people to adopt that technology because we weren't beholden to what we had written a year before when it didn't exist. And so that's what Rick's talking about here. That is just essential and so special to HudsonAlpha.
Unfortunately, it's expensive.
Yeah, I'll echo all of those things, and also just say that, when we say, grant funding doesn't cover everything, that can be a little bit, like, well, why not just write another grant, but there are some key things, and some of it has to do with continuity as well and the environment. So when we're reviewing grants, which I'm actually doing right now, you evaluate the environment of the institution, the people who are contributing, and we're going to have a lot better chance of getting grants if we can already say we've got all the equipment that we need. We've already got these people employed, and these are their names, and this is their background. If you're just running a grant by yourself, sitting there with no staff and no equipment, it's not going to score well, right? And so but having those things in place and having the continuity of it is the kinds of things that can happen with philanthropic support that can fill in those key gaps that may happen between one grant and the other.
So I want to wrap us up with one more question looking toward the future. What are you all the most hopeful for about the future of neurodegenerative disease research?
So I'm slightly biased because in my work, I'm identifying new regions of the genome that can convey risk for these diseases and identifying mutations that confer risk. And so what I foresee is very soon, hopefully, we'll be able to look at your genome and say, you've got these SNPs, and the reason this one is conveying risk is because it's affecting this cell type in your brain in this way. And it's going to contribute a little bit potentially to your risk, but we understand that now, whereas before we couldn't say that. And so that kind of contributes to what Rick had said earlier about new targets. So these are new pathways and new regions that we can kind of target with therapies. And so I'm really excited about just that gain in knowledge so that whenever you get your genotype, you don't have to look at it and be like, I don't know what any of this means, and we only know what a little bit means. We're starting to put that together. And so that's really kind of getting towards personalized medicine in these kinds of really complex disorders.
Yeah, I think I think sometimes it can feel like suddenly neurodegeneration has been exploding and growing in frequency, and to a certain degree that is true. But I think part of the reason behind that is that we've been so successful in treating other diseases. You know, the leading cause of death in 1900 was infectious disease, and in the post-World War II era, it was heart disease and cancer. And while there's still a lot of research to be done in heart disease and cancer, we've made tremendous strides in those areas. And so people are living longer and getting neurodegenerative disease. And it can take a while to turn this big ship that is the biomedical research field in a new direction, but it's happening. It's happening very fast now, faster than I've seen it happen in the last decade. It's why I'm working in this field. I used to work on cancer at Merck, and this field is the exciting field to be in for me right now because there's so much opportunity and so much interest and need in this research.
So the thing that I'm most excited about, and it's definitely a part of my answer, is what Lindsay said about being able to read out genetics and have that be as an input factor to your risk. But the vision that I have is that when you're 50 years old and you go to your general practitioner, and now they've moved colonoscopy to 45, so you won't even have to worry about that, but now you're 50 and you're gonna get evaluated for neurodegenerative disease risk. They're gonna give you a blood test and say, you're low risk, let's recheck you in 10 years, or you know what, you're high risk, we're gonna start checking every 3 years. And as soon as, for example, for Alzheimer's disease, we see a hint of a signal of amyloid positivity, we're going to start you on a preventative amyloid beta immunotherapy. And then after that, we're going to start you on a tau immunotherapy, which is what is a big focus area of clinical trials right now. So that's the vision that I have for the future. And I think that we are well on our way to that. There's a test that's a blood biomarker test for Alzheimer's that already is pretty good. The kind of research we're doing is that's why we've are doing this in other neurodegenerative diseases, and also we'll do research on some of the finer points around things for Alzheimer's disease. But that is very tractable, very achievable, over the next decade or so, and I think it's going to make a huge difference for the next generation in the level of progression in these diseases that we see.
What they said. No, I'm serious. It's exactly where it's interesting because a lot of you heard me say this. I mean, we've been here for 15 years, and I talk a lot. So, where I've said I never say the word cure because I don't know if we're going to cure. I've been working on neurodegenerative diseases literally since the early 80s. I mean, it's been a long time. And it just kind of slowly, we did learn things, and then now it's like this, the knowledge. That's incredibly exciting. And I actually see, but we haven't talked about this yet, but all the stuff we do, we use the term high throughput. We do stuff where we gather lots of information in one experiment. That's what genomics is. And used to be one gene at a time, and now it's all the genes at once. We are moving towards, and it's partly the new technology, the kind of things that Lindsay talks about and others, as well as some new things that are just are just being developed where at least the knowledge collection part is going to speed up tremendously. I mean, twofold and faster in biology or something is a big deal. Tenfold, maybe even more. And it's partly because of some new technologies and the way that we're looking. So I think this upward trend, which we really didn't see 10 years ago. I'm not even sure we were seeing it 5 or 6 years ago, but this upward trend just looks like it's going to continue climbing. We're going to help be leaders in that.
Wow. I just want to thank the panel for such a fascinating discussion. The research you're doing is really impactful, and I know I can't wait to continue following the progress you guys are making in more fully understanding neurodegenerative diseases. I would like to invite Elizabeth back up to close us out.
Thank you. Am I on, Steve? Good. Okay. Thank you, Sarah, and thank you to this distinguished panel. Tell you, it makes me so proud to work here. Really does. So I imagine we are all in this room tonight because we know someone who has experienced or is suffering from some type of neurodegenerative disease, and we care about this topic. And there is nothing that I can say better than what they said. The thing that really struck me, and I've heard them say this before, is that big pharmaceutical companies look to places like HudsonAlpha for information that they can use to go make drugs. And don't we need more drugs and more treatments for neurodegenerative diseases? It's just amazing, and it's happening so quickly. And so many of you in this room have supported the Memory and Mobility Program, and we thank you. I thought Lindsay was so eloquent about this, saying how the project she worked on was directly funded by donations to the Memory Mobility Program. And so many projects, so many things we've done at HudsonAlpha, particularly in neurodegenerative disease, started as exploratory, very promising research projects that needed to have proof of concept, so that then they could be eligible for larger NIH, National Institutes of Health grants, and that's what's happened. And you all made that possible. This community made that possible, and we are so grateful for that from everyone at HudsonAlpha. We are so appreciative of that. We hope that you will want to continue to support the work that's being done here. In your chairs, you all have the literally hot off the presses 8-page updated brochure on our Memory and Mobility Program, which will just discuss many of the things you heard mentioned tonight. So take that home, look through it, keep it on your coffee table, look through it again. If you're like me, I need to read it a couple of times as it sinks in. You will also see an interest card, and the interest card is something we hope you'll take a minute to fill out. I think there's a little pencil on your chairs. It lets us know what your specific interests may be. And then we can keep you informed as papers come out or discoveries are made, or we hold other events that are related to topics that you're interested in. So if you take a minute and fill that out and hand it to a member of the advancement team, which, by the way, if they've been saying Elizabeth, I'm Elizabeth Herron. It's my privilege to lead the advancement team at HudsonAlpha. And in the back of the room, I see Jeff Moore, Jeff Wave, Erica Gaddis, Alyssa Makowitz, Liz Wright is back there too. And just hand them the cards and they will be glad to take them. Also on your chair, of course, is an envelope. We hope that you will consider making a donation to support the Memory Mobility Program. And then we'll come back next year and we'll tell you exactly how we've used that donation by giving you another update. You can certainly mail that in. You can give it to one of our team tonight. You can also go online if you'd prefer to do that to make a donation. So with that said, let me say thank you for coming tonight. It was great to see you. What a wonderful crowd we have. We hope to see you again at our next anniversary series event, which will be on August the 17th, and that's going to be a special presentation by Dr. Neil Lamb, HudsonAlpha's president. You'll probably get an email about that soon, but we hope to see you then. So you all have a wonderful evening, and thank you again for coming here and for supporting what we do.